So chapter 11 deals with how cells communicate with each other inside of the body. But chapter 11 goes into a lot more detail than we're going to cover it in. So I would make sure to use these lecture PowerPoints as well as the chapter outlines in the study section to kind of narrow down the areas that we're focusing on and not study every single detail in the textbook on chapter 11. So just as it's important for organisms to interact with each other in the environment, it's also important for cells to be able to interact with each other and send and receive signals, either inside of a body for like a human that's a multicellular organism, or even for bacteria that are single-celled organisms that live in the same area to be able to send signals to tell them who's there, how many individuals are there. These signals are typically chemicals, so we're going to see cells releasing chemicals outside of their cells, and then those chemicals being picked back up again by cells either right next by or even very far away. Cells have to send signals to display lots of different messages. Messages for cell reproduction, for growth, for development, as well as cell death. This is how areas of one part of the body coordinate with either adjacent or really far away sections of the body. One thing that's really cool that kind of shows the unity in the diversity of life is that some of the same signaling mechanisms show up in lots of different species or in lots of different processes in the body. So we believe that cell signaling began or evolved with single-celled organisms, which hopefully makes sense if an individual cell is an individual organism and it wants to be able to communicate with individuals near it. It needs to be able to send some kind of signal. So this has been studied in yeast. So yeast is a single-celled fungus, Saccharomyces cerevisiae. This is the kind of yeast you can go to the Publix and buy to make bread with. And it doesn't have gender like male or female. Instead, we assign mating types. So there's mating type A and then mating type alpha. So this is alpha. This is a Greek letter. So if you're a single-celled organism, you need to be able to identify an opposite mating type so that you can reproduce and have genetically distinct offspring rather than just cloning yourself. So what happens is if a mating type A comes into contact with a mating type alpha, they can share these mating factors. So the chemical signal is the mating factor, and the signal has to be received somehow. So this is going to have a receptor. The A yeast has a receptor for an alpha mating factor because A doesn't want to mate with A wants to mate with alpha. And so they have a receptor for the opposite mating factor, just like the alpha yeast has a receptor for the A factor. And so you can see that the shape of this molecule is a sphere, and that fits really well into that receptor. So the way that a receptor knows if it's receiving the right signal is literally just, does the signal fit physically into that receptor? So once the signal is received, it's going to start something called a signal transduction pathway, and that's just a series of steps from the beginning to the end of where the signal was, all the way to where the signal is going. So eventually, this is a mating signal, so it's going to cause these cells to fuse. We get this A alpha hybrid, and then this cell is eventually going to kind of shuffle up the genetic material and then split back into two cells that are genetically different than the previous cells. We also see cell-to-cell communication in organisms like prokaryotes, so bacteria, and often this is called quorum sensing. A quorum, if you've ever had a meeting or think about Congress has to have a certain number of people present in order to be able to vote on legislation. That number of people present is called the quorum. So a quorum happens when a certain concentration is reached or a certain number of individuals have aggregated. Usually just means that this is kind of some minimum threshold. So this is often seen in biofilms. So biofilms are aggregations of bacterial cells. And you can see the progression of a biofilm down here from the attachment of a single cell to a surface, and then forming a single layer to multi-layers, and then eventually forming a complex biofilm, which is a structure that's kind of mushrooms up into this mature biofilm here, which has some polysaccharides, so that's going to be some sugars help holding it together. Brushing your teeth helps to disrupt biofilms. So this is an example of a biofilm that could form on your teeth. So you eat food throughout the day, bacteria in your mouth can feed on that food and then start to build up biofilms. The physical contact of the toothbrush and the floss, removing food from your mouth, disrupts this development and stops the biofilm from getting to a point where it is now going to secrete molecules to damage your teeth. Another example of quorum sensing is toxic bacteria. So often if you get an infection from a bacteria, usually the bacteria infection is not toxic, but there are some species of bacteria where when the bacteria reaches a certain level or it reaches a certain quorum of number of individuals in the body, they start to secrete toxins. And those can be deadly for specific types of bacteria. So often the use of things like antibiotic drugs are going to help kill those bacteria or reduce the number of bacteria so they don't ever reach that quorum where they would then release deadly toxins. But in order for a quorum to be sensed, all of these individual cells have to be able to tell each other, hey, I'm here, there's this many of us. And then once they have communicated with each other about the number of bacteria present, then they can start this process of forming a biofilm or secreting toxins. So there are many different types of local signaling. So local just means kind of close by or adjacent. So in local signaling, these are going to be cells that are either physically right next to each other or very, very close that are sending signals or molecules to each other. So this is often direct contact. So we've seen two examples of direct contact before. So in animals, we have these gap junctions that provide a cytoplasm to cytoplasm connection. And then in plant cells, very similar, we call it plasmodesmata. Again, cytoplasm to cytoplasm connection. There's no delay between the communication of these two cells. You don't even actually have to have a molecule leave the cell and then enter a different cell, because they can just go directly from one cell to the next. Adjacent cells are going to often participate in cell-to-cell recognition. So in one of these cells, we have a signal. And so this signal says, I am a part of my body, or I am myself, or I belong to something else. And another body cell might have a receptor that can receive that signal. And so when those cells get close to each other, the signal can bind in the receptor, and these cells can decide if they need to interact in some way. So for example, this is how we do our immune system. So our cells in our immune system, like our white blood cells, have the ability to check our cells to make sure they belong to us. If they find a cell that is an invader, like a bacteria or a pathogen, they recognize that that cell doesn't belong here, and then they can go about killing it or getting rid of it. Sometimes cells are not in direct contact, but they are right next to each other. So para means around or near. Paracrine signals are going to involve secreted messengers that are going to have to be released from the messenger cell, but they're only going to travel a little bit. So you can see that this is a messenger cell. It's secreting these green messengers, and then the target cell is the one that's going to receive the signal. So this target cell is going to have some kind of receptor that can bind these little green dots, and then it's going to receive the signal, whatever signal this regulator is giving it. One example of a local signal is a growth factor. So this is going to happen if you, let's say, cut your finger. The platelets that come to the place where it's bleeding to help stop the bleeding, they release growth factors, and those stimulate the cells that are right near that cut to grow and divide to go ahead and fix the issue, to replace the damaged skin cells and heal the cut. A specific type of local signaling is called synaptic signaling. So this space right here is called a synapse. And this is between a neuron, so this is specifically a nerve cell, and a target cell. The target cell is either going to be another nerve cell, a muscle, or a gland. So it's either going to send a message to send more messages, or just contract a muscle, or release some kind of secretion. What will happen is an electrical signal will be passed along this nerve, and then when that signal reaches some threshold at the end of the cell, it signals a release of neurotransmitters. And neurotransmitters are just like these regulators. They're chemical signals that go from one cell to the other cell, and then of course the target cell will have receptors to receive those neurotransmitters and then go ahead and continue communicating whatever signal this nerve was sending. In large multicellular animals like humans, we have to be able to send distances a long way throughout the body as well. And so we do this with long distance signaling. This is called the endocrine system, and it's going to use special chemicals called hormones. Hormones are either going to be proteins or steroid lipids. You have specialized cells within our endocrine system that release hormones. So things like your pituitary gland, which is going to release the massive majority of hormones that control the rest of your body, is a teeny tiny portion of your brain. Those hormones then get put into the blood, and they have to travel a distance to a target cell. So the target cell is no longer just right next door. It's much further away, requiring an entire transport system known as the circulatory system to get it from one place to the next. Hormones are going to be kind of a broadcast symbol. So if a hormone is transported to your blood, it gets transported to everywhere in your body. So how does a target cell know if it's supposed to react to a hormone or not? The only way to know is if you have the receptor. So if this blue hormone is, let's say, coming from the pituitary gland in your brain, and maybe it's releasing something like follicle stimulating hormone, which is going to stimulate your monthly menstrual cycle to go ahead and start developing a follicle for ovulation to begin. So that has to travel, if you're a female, all the way from the brain to, in this case, the target cell would be the ovaries. So the ovary is going to have receptors for the follicle stimulating hormone. And when the ovary receives that signal, it's going to go ahead and start the process of developing a follicle, getting ready to ovulate that as an egg, which either will or will not get fertilized. The reason the rest of the cells in your body don't respond to follicle stimulating hormone is because they don't have the receptor. So other cells that are not the target do not have the receptors. And so they don't receive the signal from the hormone, not because the hormone doesn't get to them, just because they don't have the right receiver for that hormone. By the way, in males, the follicle stimulating hormone, the target over here would be the testes, and that would stimulate the production of sperm in the male body. So now we're going to look at the 3 steps involved in a cell signal. The cell receiving a signal, the signal being passed from the cell membrane into the cell in a pathway, and then the way that the cell responds. Again, chapter 11 goes into a lot more detail on each of these steps than we're going to, so please use the chapter outline that I posted or this PowerPoint as a guide for what things I'm going to ask on the test, and if it's not included, you can assume that that's not going to be on the test. So in order for a cell to receive a signal and then have some kind of response to that signal, it's going to follow these 3 steps. First, the signal has to actually be received. So this is called reception. The reception of a signal is almost always going to happen at the plasma membrane. There are some exceptions to that, of course. So the plasma membrane has this transmembrane protein here that is a receptor. That receptor's got some sort of receiving space right there. And then here's my signal molecule. Oftentimes the signal molecule is called a ligand. The word ligate means to bind. So a ligand is going to bind into this protein, and that physical connection, the fact that this little ice cream cone fits in this triangle spot right there, is how it receives the signal. If a cell doesn't have the proper receptor, it's not going to receive that signal. Once the signal is received, it needs to be transmitted. So this is the process of transduction. Remember, trans just means across. So this is how we're going to get the signal from wherever we received it to wherever we need it to go. And this is going to occur in a series of steps. So you can see that the receptor is going to send a signal to enzyme number 1. Enzyme number 1 sends a signal to enzyme number 2. Number 2 sends a signal to enzyme number 3. 3 is a fairly short pathway. I would expect to see many more steps involved in this as well. And as each of these steps occur, the signal is moving physically closer to whatever response it's going to cause in the cell. The last step of cell communication is the targeted response. So each specific signal is going to target a specific response, and that response is going to be different depending on what the signal is, what type of cell is receiving it, maybe the environmental conditions inside of the cell. And so eventually the last step of the pathway is to send the signal to, sometimes this is the nucleus, and it's going to signal for the cell to start replicating or to turn on and activate a specific gene to make a specific protein. It may even signal to the cell that it's programmed for death. So there's lots of different types of cellular responses to signals. All right, so now we're going to start with step 1, and we're going to look at some different types of receptors. The relationship between a ligand, which is my signal, and the receptor is extremely specific. So if my receptor protein is shaped like this, it's not going to bind a ligand in a circle shape. It's only going to bind a ligand that actually fits into that specific space. So this is very, very specific, just like enzymes. When this happens, and this actually binds into the receptor, oftentimes this receptor is going to change shape. So it might have a bulge that comes out here, or it might just have a slight change of shape on the other side of the cell membrane, which is going to go ahead and kick off the next step. The vast majority of these, again, are going to be on the membrane. There are a few exceptions that are going to be found inside of the cell. So now what we're going to do is go through several different types of receptors. So for each of these receptors, I would highly recommend making some kind of flashcard so that you can remember the receptors in case there's a multiple choice question or something about them. So the first and most common type of receptor is called a G protein coupled receptor. So I know this is a long term. Let's look at the words. Receptor just means I'm going to receive. A couple just means two, right? There's two things together. And then this is the thing we probably don't know here, which is a G protein. And a G protein is just a specific type of protein that exists inside of the cell membrane. So here is my G protein. G proteins, instead of using ATP, which is adenosine triphosphate, they use GTP, which is guanine triphosphate. So it's the same basic structure with 3 phosphates, but it's got a slightly different nitrogenous base, so it works fairly similar. And that's part of the reason these are called G proteins, is because they use GTP instead of ATP as their energy source. So a G protein is coupled or paired with a membrane receptor. So you can see in this image, here's my receptor here. It's got a space where it can receive a signal. And my G protein is right next to it. It's not physically contacted. This G protein is inactive. When the receptor receives the signal, so the ice cream cone fits into that spot, what happens is it changes the shape of the receptor, which is going to trigger the G protein to go ahead and use a GTP. That GTP is going to change the shape of the G protein, which is going to cause it to move inside of that membrane. And then it's going to activate some sort of next step. So this next step might be the first step in the transduction pathway. So this is a very generic G protein image, but G protein is the largest family of cell surface receptors. So these are the most varied. They have the most different pathways that they're associated, and these are the beginnings of a very large group of different signal pathways. Another type of receptor is called a receptor tyrosine kinase. So let's talk about this word here, kinase. ASE means that it is an enzyme. So an enzyme is going to be a protein that is going to catalyze a reaction. A kinase is a specific type of enzyme that adds phosphates. So a kinase is going to take a phosphate and stick it on another molecule. Oftentimes, moving a phosphate is just enough to change the shape of a molecule and activate it or turn it on and trigger a response. So these are typically going to use ATP, and the kinase is going to take that ATP and give it to some other protein. These can often trigger multiple pathways at once, so it's not always going to be a linear path through the cell. A single receptor might be able to trigger 2 or more different pathways. The key characteristic of these that's very different is that they form a dimer. So here you can see there's kind of 2 halves here. Here's one receptor tyrosine kinase, here's another one, and they are separated here by physical space. Once those both bind to the signal molecule, so it actually requires 2 signals here, they're going to come together and form this dimer. So remember, di means 2, so that's just 2 molecules stuck together. Once the dimer is formed, then the kinase thing happens, and they're going to pull phosphate from ATP and stick it onto the tyrosines. And now that the tyrosines have their phosphates, their shape has been changed, and it's able to go ahead and activate some kind of cellular response. And so in this image, you're seeing that it's got one pathway that's going to go this way and another pathway that's going to go that way, and probably end up with 2 different cellular responses to this one signal. These receptors are misfunctioning or they're mutated. These can often be associated with different types of cancers and cell cycle regulation. Another type of receptor is called the ion channel. And so we already learned about ion channels in passive transport. So this is going to be just a membrane protein that's going to function in facilitated diffusion. So a ligand-gated ion channel. So the ion channel is going to allow a specific ion to move through it. So if this is a sodium channel, then it's going to allow sodium to move through it. This gate here is just like the door. It opens when a ligand binds. So think of it like a door that requires a key. So whenever the ligand, which in this case is probably sodium, or it could be a different signal molecule, whenever that signal molecule binds to the receptor, what's going to happen is it's going to open this gate, and then once the gate is open, the sodium is going to be allowed to move. It is able to close again, so if you were to remove the ligand, it would change its shape back to its original closed shape, and then that channel would be closed. So here's an example. Here is an ion channel. Let's say that this is calcium. So some signal molecule, we've got our ice cream cone here, is going to fit into here. And once it does, it's going to cause this receptor to change shape and open this channel. And once the channel's open, the protein doesn't actually have to do anything. The calcium can just move from an area of high concentration to an area of low concentration until it reaches dynamic equilibrium, or until this ligand is removed, and once that's removed, it will close up again. And so the thing that causes the cascade now is this influx of ions is now going to trigger some site of pathway or cellular response. Here's our last type of receptor, and this is an exception to the rule of having to be on the membrane. So intra means inside. So intracellular means it's inside the cell. So an intracellular receptor is going to be found either in the cytoplasm or in the nucleus. It's pretty much going to be anywhere inside of the plasma membrane. And these are going to be either small or hydrophobic chemicals that are able to easily cross the plasma membrane just using simple diffusion. So in this example, we've got aldosterone, which is a steroid hormone, very similar to testosterone or estrogen. in shape. And because it's a steroid, it is nonpolar, and thus it is easily able to pass that plasma membrane. It's not very large in particular. And then what happens is it's able to find its receptor protein inside of the cell, which is going to go ahead and trigger some kind of cellular response, in this case, turning on a gene to eventually make some new protein. The second step in the signal pathway is transduction. That's how do we get the information from where we received it at the receptor in step 1 to the target in step 3, which is going to give us our response. This is often a chain of interactions. So this is going to be many individual steps where the first step causes the second step, and so on and so on and so on. So we're going to see we activate one protein, which activates another protein, which will eventually reach a response. At each step, the signal is changed into a different form. So the different form might be in the shape of a secondary messenger. It might be in the form of changing a protein's shape into a different shape. So at every step, there's some change that takes place, and then that change is going to cause the next step in the pathway to occur. One of the most important reasons for having these kind of complicated transduction pathways is that they can help amplify a signal. The word here, amplify, is going to mean to make it bigger, right? Think about an amp to amplify music. It's going to take the sound that you put into the amp and make it louder. So this is a way where cells can receive a very small signal from the blood and turn it into a very big response without needing lots and lots of signal molecules to get a big response. So most transduction pathways are going to involve what are called secondary messengers, and these are just messengers that occur inside of the cell. So the most common is cyclic AMP or CAMP. So in this image, you can see that we've got a G protein coupled receptor here. When it receives its signal, so here is my signal reception, step number 1. The G protein is the second step, and it's going to signal an adenylyl cyclase here, which again, you don't have to remember these enzyme names. Adenylyl cyclase is going to cause ATP to give a phosphate to CAM, which is going to turn it into CAMP, and this is my secondary messenger. The CAMP is going to go find a proper protein kinase. Again, kinase is going to add phosphates to things, and this kinase is going to add a phosphate to something, change its shape, and then cause additional steps. So this is just one part of the transduction pathway. Just like G protein coupled receptors are the most common and varied receptor, cyclic AMP is one of the most common and widely used secondary messengers within the cell. So it might seem a little bit overly complicated for cells to have 15 different steps that they need to go through in order to get a signal from the plasma membrane to the nucleus. Why not just send CAMP directly to where it needs to go? And part of that is because each step allows us a chance to increase the response. So here's an example. I've got one G protein. So this G protein is going to receive one ligand, right? Or this is the signal. So this means that in the blood, I don't actually need to have a lot of hormones or whatever signal molecule flooding the blood. I can have a few. So one is going to bind to the cell. So this one G protein is going to activate 100, 10 squared is 100 adenylyl cyclase, this is now the second step. Each adenylyl cyclase will activate 100 CAMP. So now we go from 100 adenylyl cyclase to 1,000, 10 to the fourth is 10,000 cyclic AMPs. So we started with one ligand and we're already up to 10,000 secondary messengers. Those secondary messengers activate one protein kinase each, so we're still at 10,000 protein kinase A. Each of those is able to activate 10 of another phosphorylase kinase, so 10 to the fifth is now 100,000 phosphorylase kinase. And then eventually it's going to reach its final step, which is to activate glycogen phosphorylase. Each one of those is going to activate another 10. So we end up with 10 to the 6, which is 1,000,000 active glycogen phosphorylases. And again, you don't have to remember the name of these molecules. The point here is that we started with one signal coming from the blood, getting to the cell. And we ended up with 1,000,000 molecules involved inside of the cell in the response. And so this is amplification, going from 1 to 1,000,000, and we only increased it in 6 steps. Having 6 steps here allows us to drastically increase the response that a cell can have to even a very small signal. And eventually we will get to the 3rd step, which is the cellular response. So we're using response here as kind of a generic term. The response is just whatever the cell does in reply to that signal. So up here we've got reception, transduction, and then in this case our response here is happening in the nucleus. And what it has done is it turned on a transcription factor, and here's my DNA, and it's going to go ahead and start protein synthesis to make some protein product. So this can happen in the nucleus. A response can happen in the cytoplasm. Sometimes this is going to turn a gene on. It can turn a gene off. It can create enzymes. It can make other proteins. Sometimes this is going to go ahead and lead to other cellular activities like cell replication. So if we had a growth factor, right, this is a growth factor, this is maybe going to go ahead and stimulate that cell to replicate, or a different signal may even signal that that cell is mutated or damaged and that it needs to go ahead and be demolished. One of the other advantages to having lots of steps in the signal pathway is that there's lots of places for variation in responses. So the specificity of the response, so what response you get from each signal, is specific to each cell and specific to each pathway. So here we've got cell A, and this is cell B. They're going to receive the same signal. In cell A, that signal has one pathway, and it proceeds to response number 1. In cell B, that same signal has a different pathway that actually splits into 2 responses, and we get 2 completely different responses from the same signal, just because these 2 different cells have a different transduction pathway, right? That's step number 2. Sometimes along these transduction pathways, so on the way to the response, you will meet what's called a scaffolding protein. So if you think about a scaffold like a building that's under construction, the scaffold is the thing that the construction workers build on the outside, and it looks kind of like bars, and they climb up it to be able to do work on the building. A scaffold is going to be kind of on the outside, and it's holding these 3 proteins together. And what's happening is these are maybe 1, 2, and 3 steps in that transduction pathway. So when the signal is received up here at step 1, that signal can go to the scaffold process, and then it can go through that transduction pathway fairly quickly. And so that's going to make transduction really efficient. And it could be just efficient because the 3 molecules are physically right next to each other, and so the pathway can go quicker. So oftentimes these scaffold proteins are found in pathways where the transduction is going to be very efficient and quick. So up to now, we've been talking about how to start a signal and how to send the signal. But at some point at the end of the message has been received, and we turn it off. So eventually the signal has to stop. And there are several different inactivation mechanisms, and these are important for cell functioning. You don't want to turn a gene on and then just leave it on. It's not like you don't turn a light on and just leave it on. Eventually you do want to turn it off. The easiest way to do this is stop sending the receptor. So when there's fewer receptors, they're not going to bind. And when they don't bind to the receptor, then the signal will turn off. The receptor will go back to its inactive state, and then the entire signal pathway will stop. So it'll unbind and revert to being off. This can also happen in the relay, so that transduction pathway. Once the receiver is off, then those relay molecules get turned off as well, and they will go back to their inactive forms, and then eventually the response that the cell was giving will turn off as well. And then it can turn back on again as soon as that signal is received again. So we've talked a little bit in general about the different types of signal responses that cells can give, and I want to talk about apoptosis just a little bit here. So say this to yourself, apoptosis. It's a fun word. And what this means is programmed cell death. So this is when a cell is killed specifically because it needs to be killed or it's time. So what happens is in this cell, the components inside of the cell are chopped up and put into vesicles. And those vesicles form kind of lobes, and the scientific term for that is blebs. And so over here you can see a cell with blebs. And this is kind of like an internal demolition. So what's happening inside is the lysosomes inside of those vesicles are chopping up all of the packaged pieces. But instead of just popping the cell and letting them all out, it's doing it in a way that's contained. So it stops it from leaking out because it might damage the neighboring cell. And so this way, only this cell dies, and this cell is able to maintain its proper function because this cell is going to be demolished internally and not leak out or damage its neighbor. Apoptosis is common. Everybody has done this, and this is a normal part of life, is killing off cells that are damaged or cancerous or just are part of proper development. So if you look at this down here, this is an embryological hand between about 7 to 9 weeks of development after fertilization. So a baby's hand starts out as a nub, and you can kind of see that each of these little bumps is going to be a digit, and there's membranes in between all of the fingers. So eventually, the cells between the digits start to go through apoptosis because we've evolved to have very dexterous and flexible fingers, and we don't actually have membranes between them. Eventually, in a week or two inside of the womb, this actually happens fairly quickly, the spaces between the digits are clearly defined and none of that webbing remains. So if you know anybody who has webbed fingers or toes, that's just because their cells didn't properly go through apoptosis while they were a fetus. Apoptosis is sometimes used by tumor suppressor cells to contribute to killing cancer. And so if apoptosis is stopped or it's not properly brought about, then that's how some cancers are formed. So a cancer would be a cell dividing when it's not supposed to. And if that cell was supposed to be killed and then wasn't killed, it could continue to go on and form some form of cancer.